Good morning, my fellow scientists. It is Wednesday, June 21st, 2017, and I want to talk to you about alpha-galactose. Alpha-galactose is a saccharide found on animal cells, but not on human cells, and our immune system recognizes it as foreign. I just read an article about an AlphaGal allergy, where the immune system becomes hypersensitive to AlphaGal after a human has been exposed to a tick bite. So I've put a link to the article down in the description. It's at wired.com. It's really interesting. There's an unknown element in the tick bite, possibly due to the tick's previous meal, that causes the human immune system to go crazy over alpha-gal. And as a consequence, when that person then eats red meat, their immune system detects the alpha-gal in the meat as being foreign and has a huge allergic response that can make people very sick. So biochemically, what's happening here? What is it in the tick that makes this happen? And how can we interfere with this process in order to allow people to eat red meat again? I thought that was really cool, but it reminded me of a TED Talk that I heard some years ago by a guy named Kary Mullis. Now, Kary Mullis is a pretty famous scientist who invented the polymerase chain reaction and won the Nobel Prize, and then went on to found this company called Altermune. Now, Altermune uses the body's innate immunity against alpha-gal as a way to flag the immune system against things that we don't want in our bodies. So in his TED Talk, he talks about how he had a friend who died from a infection with a drug-resistant bacterium, and he wanted to do something about it, and so he selected an aptamer against these drug-resistant bacteria. An aptamer is a little chunk of DNA that folds up and binds to a target. So once you have an aptamer that binds to a target like one of these drug-resistant cells, you can then chemically add the alpha-galactose to the end of the aptamer. Now the aptamer is sort of a paint that tells the immune system, the thing I'm bound to is bad. And he started a company that made these Alphamers, these alpha-gal bearing aptamers, in order to inspire the immune system to kill things we don't like drug-resistant bacteria or potentially cancer cells or anything else that we can select aptamers for. So I think that's a pretty cool technology. I hope it works and becomes very successful. It turns out this year, in January, Altermune sold the Alphamer technology to another company who hopefully will be able to commercialize it rapidly. So I think it's funny how two very different subjects, the epidemiology of allergies and the biotechnological solution to drug-resistant bacteria, can end up having this sort of common root in this immune reaction to a strange little molecule called alpha-gal. Anyway, links in the description below if you want to read more about antibodies or allergies or alpha-galactose. If you like hearing about this sort of thing, chemistry, biochemistry, aptamers, and future biotechnology, tune back in tomorrow. We update Monday through Friday. We discuss all kinds of interesting science here in the Allen Lab.